seperti dia lagi, mau tahu nih dia loh. Hah? Tuh mau mungkin waktu. What is it? Come on. What the nose cut? ini berat, saru presentation tuh pengen kayak gitu bukti kori. Nah, dia ada di tes lain tulisan jauh yang tulisan itu di bawah. Bisa, itu berat. Bisa?
Juvenile port,
Itu juga dia nah itu pentai itu pengen kok sari di tes Korea itu saya sudah lihat ya kalau orang ini.
Oh,
Itu boleh dia.
mana?
Mana
Online ini itu sorry.
online itu sudah dapat harus ini দেখা পাওয়ার নামে প্রেজেন্টেশন তুই দেখা পাবি মানব দেব আমি ইয়ে দেব no no ini মানব হয়েছে মানে জীবন মানে আমার সব বাউপি কোরিয়া itu because the local job for ya kan sama nuaru asal tidak boleh asal tidak boleh ini saya kan sangat paling itu mana kuai mana itu kalau saya network connection baru itu mana display dokumen oke terus saya tuh testing kalau oh boleh coba kan DJ paling dia dari sana oh itu অন্তত যদি প্রেজেন্টেশন হেতুতে এসেপ্ট করব লাগবে নাকি যদি প্রেজেন্টেশন দিয়ে হেতুতে এসেপ্ট করব লাগবে নাকি বেয়া এসেপ্ট করব লাগবে কিন্তু তখন তাই মানে কি আর জয়েন করছ না তুমি কি বলে এর আর পড়ে গেল লাগে তো আপনি তো মোবাইল আপনি তো এই অর্গানাইজেশন মোবাইল মোবাইল Hello?
A very good morning, everyone present here. On behalf of the organizing committee, I would like to welcome you all to the DBT-sponsored two-day national seminar on recent advances in applied biology, organized by Eco Club in collaboration with IQAC and Tihu College Science Forum, Tihu College. I'm glad to announce that we have our chairperson and invited speakers, so we are ready for the session. I would just like to welcome our chairperson, sir, and invited speaker, sir, and thank you so much for giving your time. As the chairperson of this technical session number 7, we have Dr. Anil Boro, sir, who is the associate professor and HOD of Department of Geography, Tihu College, and he is also presently the IQAC coordinator. As the invited speaker of the session, we have Dr. Kuladip Sharma, sir. Dr. Kuladip Sharma, sir, teaches animal ecology as an assistant professor in the Department of Zoology, Guwahati University. His study area focuses on how plants and animal communities respond to their abiotic environment, examining the effects of habitat alteration in multidimensional climate space in eastern Himalayan region. Sir is also curious about the extent of species conservation in the interaction between human and forest. His research area and of interest are as follows. It is theoretical and applied ecology, mathematical ecology, and conservation science. Sir has also completed various research projects funded by Rupert Foundation UK, Conservation Leadership Program UK, US Fish and Wildlife Services USA. Sir also has a lot of projects going on, and sir has n number of research articles which is impossible to sum up in such a short period of time. So thank you so much, sir, for your time. Now I would like to request Rita Devi ma'am to please felicitate our invited speaker, sir.
Yes, dear. রবীন্দ্রনাথের কথা আপনি মোবাইল itu tadi itu jawab terus dulu kita sudah enggak itu sistem hybrid apa kalau kita kalau
Good morning all present here. Today we are second day of the CBD Sport for National Seminar, and this is the seventh technical session. And now as part of schedule we have the first invited speech. The study is already with us. The topic of this speech is importance of biodiversity measurement, understanding of rare and common species. By Dr. Kuldeep Sharma sir from Guwahati University. I request invited sir to present his speech.
Thank you very much. Thank you, first of all, for inviting me, the organizing committee. And I'm very happy, I'm glad that I'm here in Tihu College for the first time ever. Not been here earlier. So, I should start with my presentation, which is the importance of biodiversity measurement, understanding rare and common species. Actually, it's a kind of a campaign that I and my lab mates we have started very recently. This campaign is about understanding the importance of data in ecology. Most of the time, what happens in biological science, we are kind of deprived, or not deprived, we are kind of discriminated by other science disciplines because we have been considered as a less accurate or kind of a hate type of science because we write a lot and our data part is missing somewhere. So we have started this journey as a ecological data science, and as a part of that data science, we have started this campaign called importance of the measurement. How the ecological parameters should be measured and how we should approach the ecological studies. So, as all of we know that what is biodiversity, we need not to explain it. So many things are there, and most of the time we are really very happy by looking at those pictures, but not the data behind those pictures. Like nowadays, big data is coming. So in big data, we can extract so many information from those pictures. But in ecology, or most of the non-ecological, non-biocentric, they are also very happy by looking at those things. So it's kind of irony. Next, please. So what is the uniqueness about ecology? Ecology, or I should call it as a biological science. It's a multi-causal system. It is not like physics, not like chemistry. Two chemicals, chemical mixtures will provide or give you a particular color. It's a multi-causal system where so many factors are affecting, so many factors are working to give you the result, to give you the response. So please. Go back. Yeah. So we rely on most of the time we rely on the qualitative approach. Qualitative approach means whether number of species are more or less, or disturbed or undisturbed. So those are some qualitative approach we have been taking up. Then, as I have already mentioned, ecological data often questioned by other scientists because we do not have a robust way of collecting data. We do not have a robust understanding of the methods, whatever methods we are using. Next. So. Next, please. So next thing is that what are the challenges? Challenges which what challenges we face? I think it's okay. Slides are not that important. We will talk that is more important. So the next thing is the challenge. What are the challenges we face while collecting data or while thinking about the robustness of the data? That is why that part is important, because see, when we collect data without a proper method, if we collect data from the field and come to the lab and say that this is the condition of that species, this is the condition of the area in terms of biodiversity, then we believe that, or we expect that everyone should believe us. Everyone should believe us without questioning the methodological approach or methodological part, which is kind of ironic, because that is not science. If we see in a broad spectrum, that is not science, because science should be repeatable. Once the other researcher, if he goes there, he should get the same kind of data, same kind of parameters at that place. But that is not happening. Why that is not happening? Because somewhere in our data collection, somewhere in our data interpretation, somewhere in our data analysis, we have been missing a particular robustness point, or we have been missing particular analysis accuracy in the analysis of the data. So that somehow we have to brought those missing points into the picture. So how we are going to do that? That is about ecological data. And most of the time when our students come to us and say, like, sir, I want to study biodiversity of a particular area. That's very our notion is that, okay, you are going to study biodiversity. It seems very easy topic. Okay, biodiversity. Okay, you will go to some forest or some place and you collect whatever there in that forest or in that area. So that should not be the case if we see the approach in a very scientific way. Because there are so many indices available. There are so many assessment available by which we can assess whether a particular study is accurate or not. That accuracy should be assessed in a very scientific way. And in that way, if we think about biodiversity assessment, if we think about different types of indices that are available nowadays, those indices are very, very like old indices. Most of us, we know about phenomenal index. Phenomenal is the diversity index. So that index is described around 1891, which is still we use that, and nobody is questioning that. Why that index is so robust, or why that index is so widely used. So we are not questioning because we are not interested in knowing how to measure biodiversity. So that part we are now taking up, and one of my research scholars is doing research on that, how we can incorporate some other regional parameters in the diversity index, like to make it more robust, to make it more. So that please. So we have a limited quantitative understanding of ecological parameters. That's what I have explained. Like, then robustness and ecological contributions are not very robust. Next, please. And obviously, next back again. Obviously, we are seeing this picture. Obviously, we are very much interested in photogenicity of the animals. If we post a very good-looking, cute animal, then obviously our study is complete. Like, okay, he has presented so many good pictures. So that's how we our perception is about it. Next, please. See, there are various patterns. There are various patterns. This is one recent paper we have like communicated. So one of my intern, she has done this in Guwahati city, urban city. So there are various patterns. There are around seven different places in parks and urban green spaces. See this pattern. This is species rank. This is relative abundance. Most of the time we are considering numbers, but numbers are not enough. Like this species has this many numbers, this species has this many numbers. But numbers are not enough because unless and until we know the relative number, it is useless. So if we plot the relative number, we could see the arrange them in descending order. Then we could see that the first rank and second rank, the difference is so high. Some cases, like there is only one species which is having so many number of individuals, and just next to that species, the other species are having less number of individuals, which is very like amazing fact in biodiversity. Like one species is having so many numbers of individuals, and other species is having so many less from the top one. That is called rank abundance curve. And while studying ecology, we are making it complex by giving some jargon or giving some name to these graphs. Like this graph is known as rank abundance curve. Why to call it as a rank abundance curve? This is simply a pattern of biodiversity. This is simply a pattern of biodiversity. Another very easy and simple pattern is this. Like when we go to a forest, we could see the number of species which are rare. Number of species which are rare are always more than the number of common species. Like if we see in and around, then we can see that some of the birds are very common everywhere. You could see so many number of individuals of those birds, like common myna, halika. Then there are so many like dove, spotted dove, and all those birds that are in and around us. So the numbers of those birds are more, but the number of species of such birds are less. That's what this graph is depicting. So if one student of mine, he went to or he goes to somewhere collect biodiversity data. If I check the data in terms of this graph, because this is a generalized pattern, then I could see or I could clearly like validate his data by looking at this graph. So this should be the original pattern. Next, please. See, another example is that if we are considering two communities, community 1 and community B. See, in both the communities there are two species. But here in the first community, the species 1 has one number of individuals, species 2 has 19 number of individuals. But in the second community, species 1 has 19 number of individuals, species 2 has one number of individuals. See the compositional difference. Like if we see the species richness, both the communities are same. There are two species only. Both the communities are same. But see the number of individual in the perspective of number of individuals. So in one community there is species 1 is 1, in other community the species 1 is 9. So obviously the resource wise, the functional area wise, both the communities will behave differently. Like their response will be very different. Though we do not study that part while studying the biodiversity. That's where we are missing the point. Next, please. So this is very important. As I have mentioned, data collection. How we are going to collect it? How we are going to collect the data and how we authenticate the data. How we authenticate data means like if one of my students is working, like I should be very precise while collecting the data, not while analyzing the data. Analysis is very like a post-processing thing. So during collection of the data, we should be very careful like what data you should collect and how you are going to collect that data. And obviously we have so many tools and tools available. Like this is a handheld GPS. There are other tools also. Like one is range finder is there to see the micro habitat and all, height and all. Then this is very important. Like how we interpret the data. Suppose I always give to this example I always give to my students. Suppose if we think that all the species are, if I put all the species in a glass, hypothetically, of a particular area, all the species, if I like number of individuals, all number of individuals, if I put in a glass, then when randomly pick 50 out of that glass, how many species would be there? Because I am collecting 50 individuals from that glass. How many species would be there? So that is for rarefaction curve. Rarefaction curve says that like every time we sample, every time we go to the forest, every time we collect data, how many species we are expecting? Like number of individual wise, how many species we are expecting? Suppose I am collecting 100 individuals or I have observed 100 individuals today. In these 100 individuals, how many species be there? Like in terms of species. So that is not that complex. But when we see the graph, it appears that it's very complex. We should not doing all these things unless and until we are very good in statistical analysis. We are very good in that in this. So these basic patterns are very important for all the researchers, for all the faculty members also, and that's why this campaign is very important. Because unless and until we make it a very widely spread campaign, we won't be that much of serious about biodiversity. Next, please. So in conceptual mode, we can say that the landscape parameters set biodiversity. As I have mentioned that we have been using that non-linear indices for so many years. But in that index, if you see, there is no landscape parameters considered. In non-linear index, only two things are being considered. One is the number of individuals, other one is the number of species. So the landscape parameters are missing. Then reinterpretation is very important. Like we have indices, we have so many things, so many statistical tools are there, but the reinterpreting those indices, or kind of incorporation of some new parameters into those indices is very important. Then this is a debate. This is a very highly rated debate. One is called presence, whether presence is enough or not. Like if we say that particular species is present here, is that enough or not in biodiversity assessment? It's a very pertinent question. Only presence is enough or not. Next is the relative weightage of species matters. Relative weightage of species means if there is a tiger and there is 100 elephants, see the relative abundance of these two. One tiger, 100 elephant. Then again 100 elephant and suppose 5 deer species or 5 deer individuals. Then obviously the relative abundance is playing a role, because 100 elephant will consume so many resources which are also shared by the deer species. So accordingly the whole community scenario will be different. Next please. So this is a case study. Because I have only 30 minutes, so I am not going to explain everything. So this is a case study, as I have mentioned already, that one of my intern students, he has done imaging work on urban green landscape. She has identified so many urban green spaces and urban parks where she has studied coleopteran and hemipteran species. See, in this case also, my lab, we are not interested in the group, the taxon. We are interested in the number of species and the individuals, not on the taxon, because these are models. We can replace this model by some other individuals. And next please. So the goal was previous one. Okay. The goal is identify the most ecologically appropriate representation. So her dissertation goal was to identify the appropriate presentation, how we are going to present the biodiversity. Next please. Next. Okay. This is very interesting. This is called Christian model. Christian model is one of the least used model in biodiversity assessment. What it does, this is a log normal distribution. Log normal distribution means the x-axis is log base to converted. Log base to converted means the abundance means number of individuals are converted to log normal distribution. And in this log normal distribution, it should be conventionally it should be a normal distribution. Normal distribution means a bell-shaped curve. It should be a normal distribution. But see, we are seeing here only one part of that normal distribution. That means the other part is the rare species. In this quasi-urban landscape, we have not encountered or have not recorded so many species because this curve is like this. These much species are missing. These much species are missing. That means our sampling is not whatever sampling or whatever sampling effort we have done we have conducted is not enough. In this case also for Coleoptera and Hemiptera both, the log normal distribution shows that is not accurate. But see, most of the ecological papers, most of the biological papers, this kind of interpretation is not there. This kind of interpretation is not there, and nobody like us. If I with a reviewer, I am also not going to ask any questions about the number of species or the number of individuals that you have missed during the survey, which is very important because we are finally saying that these many species are present here. Suppose in Manas National Park, these many species are present, but that may not be true because the surveyor, the observer, he might miss so many species, and this graph will tell you how many species that surveyor this researcher might have missed. So this is how we should tackle the problem. Should handle the problem. This is a model we have developed. In my lab, we have developed this model. In this model, we are trying to map the richness in a spatial map. Spatial map means this is a spatial map. In this map, see the richness is higher in the red, dark red circle or dark red polygon. And why this is important? Because this will tell you that the species richness or the probability of finding the species or the potential of finding the species is lesser and lesser if we go outside from that circle, from that red circle. If you go outside and outside, the probability is lesser. Now the major question is whether we are interested to give some indices which is based on the data, and that data is not complete yet because the observer has missed so many species, or we should give an overview of that area where from we can say that okay, this area might be occupied by such and such species, and the probability of that occupancy is lesser and lesser outside that periphery, outside that circle. This is how probably, because this is not yet validated, and one of my research scholar is doing his PhD thesis on this. So if once his PhD is like approved, then we can say that this is what we can do instead of doing some like inaccurate assessment in ecology. Next, please. So call for action, as I have already mentioned, we are running this campaign. So make ecology a more robust scientific discipline. This is our aim. Next. So biodiversity studies must go beyond description, like beyond the species description, like these are the species found here. This should not be a scientific approach to explain biodiversity data. Next, please. Obviously, we are not explaining any complex parameters, complex indices. Some simpler diversity patterns offer meaningful insights that you often miss, or that you often we do not give those explanations. Next, please. Yeah. young ecologists or young those who are interested in ecology. They should take studies with greater seriousness. That seriousness is highly. Good. Next, please. It's the end of the presentation. Next. Yeah, this is my team. Very young team because I have joined 4 years back to our university. So this is my team, and we are constantly trying to analyze data and see how the pattern of data can help us. And all of you are most welcome to my lab. It's a quantitative and predictive ecology group. We are a group, not lab. And all of you are most welcome. At any point of time, if you find any data, then you can contact us here in our lab in the second floor of the department. Thank you very much. I'd love to answer if you have some specific queries.
Thank you, Sharma sir. It's very nice presentation and good presentation. Now this open for discussion. Any question? Question.
First of all, thank you for your valuable insights. And if you remember me, but it was before 2 months that quiz. Third, since I'm carrying out my research in entomology, particularly the technical right, I'll be definitely glad to join you, considering your last slide. And I have a question, sir. What are the sampling techniques that are commonly or used in recent times for study of insect species, various species of insects, be it related to vector biology or related to agriculture?
Thank you for the question. It's a very common or widely asked question, and
Lots of biases.
yes, yeah, biases.
No.
That's why I always in my class also I always used to confuse my students by saying so many things together. So I am going to do that again today. So when you are looking at some species as a part of your PhD program, then you should not thinking about any other methods given by other scientists, ecologists. What should be your aim? Your aim should be how am I going to represent the whole study area of your interest by taking enough samples. That is the primary thing. So, see, there are so many methods that are written on papers. Papers mean scientific papers. Most of them are vague or most of them are incorrect. Most of them are incorrect, unfortunately. So, what we should do, we should be thinking about dividing the whole area, whatever area you are surveying, dividing the whole area into some approachable sampling sites. And within those sites, you can apply any method. Like suppose you are working on spiders. If you are working on Hemiptera, if you are working on Coleoptera, then obviously you can do random search. Because the Coleoptera in a line or in a transit walk, you may not see a Coleoptera. So a random search is good. But where you should start that random search and where you should end that search. That decision you have to take. And once you have taken that decision, if you mention that decision in your method part, then it is a valid method. If you mention that part, like I have started here and I have ended here, and the starting time is this, the end time is this, then it becomes a valid method. Because the next question the reviewer will ask, or I'll ask to you is like why this method? Then you should be answering like, okay, because I want to cover whole of my study area, or I want to have representation from my all parts of my study area. So I have divided it in such and such way. And from each division or from each side, I have taken samples, which is very important. So random search is one, which is like widely used by researchers, but the earlier part is not explained by the researchers. Like what is the way where he has done that research? Getting my point? Thank you, sir.
And just small clarification. regarding the biodiversity measurement, you have mentioned in your presentation that there is no robust method for the collection of these samples and these species. Do you think that our national parks like this Manas or Kaziranga, the data provided now available still on that is incomplete?
Yes, sir. Yes, sir. I like strongly feel those data are incomplete. And I always contradict them also. Like now we are saying that the number of tigers is increasing. Number of tigers is increasing. But see the methodological history of tiger census. Earlier we conducted tiger census based on pug marks. Now we have camera traps. So obviously camera traps are more efficient than the pug mark survey. So now we are getting more and more tigers. That may be one reason. Unless and until we are very sure about that evolution of the tools. So we cannot say that tigers are increasing. There may be so many more tigers or less tigers in Kaziranga National Park. So the list is never complete. Like the whole list is incomplete because unless and until we validate the data using that Christian model or some kind of robust statistical technique, we cannot say that those data are complete because most of the time we only think about the number of species. And this species, how many number of species are there? That's all.
Applying these available methods or techniques, how much time will take for this proper understanding of the species of the biodiversity of this?
It won't take much time. That's what we think that if we explain whatever we have done in the name of survey, that is enough to explain the data. Because most of the time we say that I have surveyed in Kaziranga National Park. We don't say that I have surveyed only Bagori range, I have surveyed only one range of Kaziranga, one particular pocket of Kaziranga National Park. We always conclude it for the whole national park, like for Manas National Park or Kaziranga National Park. But there are so many pockets. And most of the time we know only the charismatic species, like tiger, elephant, or the cat species. But we don't care about Coleoptera. We don't care about Miptera. We don't care about all the insect species that are present.
Sorry, you think that still there is much scope or much work to be done for the proper data for these our national parks.
Yes, sir. So many things to be done.
Thank you.
Thank you, sir. Thank you.
Really, good session. I would like sir to carry on this session as a chairperson, and I would like to request Ms. Rita Devi ma'am to felicitate sir with an RNI. Thank you so much, ma'am. Sir, it's up to you now. Session. Thank you, sir.
Yeah, I'd also like to call my teacher, one of my teachers is here, Jana, and here I can see here are there. They should join me in this. Somewhat awkward of the I should not be here. By also the university portfolio. So the next speaker is Tika Bora. She will talk about range of tradition words regarding Tika. Online. Yes, starting with the Thank you, Sika. Thank you. Thank you, Sika. Sika, thank you. Okay. Thank you, Sika. To Sika. Thank you very much, Sika. Thank you very much. Yeah, yeah, thank you very much, Sika, because you were assigned only 10 minutes. Okay, you can just, yeah, yeah. My question is that what is a new thing you are explaining, because everything is there in literature, like whatever you have explained in your slides. No, no, we are getting your points. Like you have the excellent presentation. It's a very good presentation. But my question is that for this conference, what new message or what new idea you are presenting in through your slides. Yeah, now the session is open for questions. Yeah, now the session is open for questions. Now the session is open for questions. Any questions from the participants? The online participants may also ask questions. Is this a part of your dissertation work or PhD work? Silvi, is this a part of your dissertation work or PhD work? Okay. Is there any question? If not, yeah, there is no question, Sika. There is no question for you, Sika. So we are going forward. Moving on, the next speaker is. Thank you, Sika. Next speaker is first report of uh, Incorporates Humanities from Guwahati, Assam by Santana Saikia and the Girls College, Guwahati. Santana, please start your presentation. You are not audible. Santana, you are not audible. Santana, if you are having problem with your internet connectivity, then we can move forward to the next speaker. The next speaker is Kumar Kitartha Kosi. Kumar Kitartha Kosi, if you are ready, yeah, you can start. You can start. Yes, yes, very much. Thank you, Peter. It's a very nice presentation and nicely prepared, like precise and timely and completed presentation. So, first question is from my side. Why is this the part of your PhD research? Okay. Have you investigated the root microbes in both the plots? Okay, because in recent studies, most of the papers are published that microbes are like more convenient to study either growth and other patterns of crop growth maybe. Because it is a very interesting study also biochar and what is the like if you move to commercial, what would be the scale of production of biochar? Like can okay. Thank you, Peter. Thank you very much. Now the house is open for questions. If anyone has any queries from online participants, if you have any queries. Okay, thank you, Peter. Before we move forward, there is actually one announcement from the organizer side. So I request the organizer to announce.
Thank you so much. I request the participants to please stay connected with us because we have with us Dr. Pankaj Gora sir, and he will be delivering an invited lecture in this particular session. So, sir, am I audible to you? Okay. Sir, good morning. We welcome you to this EBT-sponsored two-day national seminar on recent advances in applied biology, sir. For the participants, I would like to introduce Sir. Dr. Pankaj Gora sir is a distinguished computational systems biologist and a respected group leader of Evo group at Tezpur University. With over a decade of extensive experience, his expertise spans at the field of genomics, systems biology, bioinformatics, machine learning, and predictive modeling. He earned his PhD in computational systems biology from the Norwegian University of Science and Technology. His research is centered on investigating evolutionary mechanisms in biological systems from system level. He employs modern approaches, including the analysis of high throughout the data, next generation sequencing, etc. Prior to his current role, Dr. Bora had held significant positions, including a tenure as a bioinformatics scientist at the German Cancer Research Center and a postdoctoral fellowship at the Norwegian University of Science and Technology. So we are glad to have you, sir. We can move to the invited lecture by sir. Thank you.
Yes, sir.
Yes, sir. Yes, yes. Coming, sir.
On behalf of the organizing committee of the seminar, I would like to extend our heartfelt thanks and gratitude to Pankaj Bora sir. Thank you so much, sir. It was a very insightful talk, and we are delighted to have you. It was great listening to you. If anyone has any questions, they are free to question sir on the topic. There is any question? The online participants are also free to ask any questions. You can just unmute yourself and ask. Sir, I believe there is no question. Your lecture was totally insightful and beautiful. So thank you so much, sir. Uh, The participants who have joined online, there is an announcement from our side. You are requested to please limit your talk or presentation to 7 minutes only. It will be great if you could just follow the timeline because we are already running late. So it is a request from our side. Please limit your presentation time to 7 minutes and 2 minutes will be assigned for question and answer session. Thank you. Over to Chairperson, sir.
The next speaker is Santana Saikia. I think Santana is ready now. Okay. Then should I call the next speaker? Santana, please let us know when you are ready. So we are moving to the next speaker, Rajashree Deka. Rajashree Deka? Yes, yes. Yes, visible. See, please, thank you so much on the key findings of your study. Thank you. Thank you, Ravi. Thank you, Ravi. Thank you. So, is this a published work or have you published it somewhere? Thank you. Now the session is open for question. If you have any query, any specific queries, then you can ask. No queries? Online participants? Okay, thank you. We are running out of time, so we are moving forward. The next speaker is Parmita Salma. Parmita Salma, Autodesk University. Parmita Salma, are you ready? Yeah, go ahead. And please keep the introduction and review methods apart. To directly show your objective and result.
your voice is breaking. Your voice is breaking. And Karmika, your slides are not visible. Karmika, your slides are not visible. Your voice is also breaking. No, still not visible. No, slides are not visible. And your voice is also breaking. I think you have internal issue. Can you wait for some time so that we can finish Santana's presentation? You try to reconnect. Okay. I I think Santana is ready. Santana, Santana, you are ready? Yeah. Yes, Santana. You can go with your presentation. It's okay. You go ahead. Present your findings. Yes, you are audible. You are very- No. Yes, now it's visible. No. It is not visible. It goes again. You share your slides. No, it is not visible. It is not visible. No, it is not visible. Yes, now visible. Now it is visible.
Thank you, Shantana. I think you have published this paper, right? You have published this, huh? Published this yet? Any chance? Oh, then you should not present like first report. Then we should not present it somewhere else because it's a first report. It's very nice that you got this from our teacher. And nice presentation, and thank you very much. As I know you in person, so I have a suggestion. Yeah, Santana. So as I know you in person, so I have a suggestion that we expect you to be more smarter in technological tools like in handling technology. So next time we should not feel that. You know? Yeah, yeah, yeah. Next speaker is Parmita Sharma. Yeah, you can present. Yes, basically. You scroll the slides. Yeah. Thank you, Pavela. Thank you, Yolita. Now, my question is, what have you taken this study? Like taken up, do you have any specific objective for studying the differences of the morphometric characters of this species? Because they are already recognized as separate species. So they are obviously they are having differences, right, in morphometry. That's why they are a separate species. Now again, why you are studying this? Yeah, again, then why you have taken this study? Like, do you have any other objective to, you would like to know something else about between what is there between or among the species? Okay, thank you very much. And I request you to send your slides in a PDF copy of your slides to the organizers because your presentation is not at all like kind of aligned with your talk. Like the slides were lagging behind, right? So you please send your slides in a PDF, make it a PDF copy and send to the organizers. Thank you very much. So formally, this is the end of the section 7. Now we are going to start the session 9 directly. So the first speaker is Ashish Bhomi. I think this session is also online. So I request Ashish Bhomi to present his findings. Ashish Bhomi, are you present online? I think Ashish Bhomi is not present. So the next speaker is Hamida Khatun, South Salmana College. Hamida Khatun, are you present? No? Hamida Khatun is also absent. The next speaker is Biju Borkotoki. Biju Borkotoki, I request you kindly to present. Yes, you can present your findings. No, not visible. No, 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 not yet. You click on the share button and share the whole window. Then open your presentation slides. No, not visible. On your
will be written on your screen, you see if it is written that you are presenting, that means it should be like it should be visible. You can send your presentation to the organizers. You can send email your presentation to the organizers. Then you have to present. At least you have to show the slides. If you can send the presentation in a PDF copy to the organizers, then we can see it here and we can receive. Yeah, what's in WhatsApp group also you can send the presentation. There is one WhatsApp. Okay, we are moving forward. We call the next. You decide, please. Each of board of please you decide what you are going to do, like whether you want to submit or not. Then the next presenter is Nayanika Devi, University of Science and Technology, Meghalaya. Nayanika Devi, represent. Yes, yeah. Please present your comments. Biju Gaupati, please mute yourself so that Nayanika can present. Yes, Nayanika. Yes, Nayanika, you are very much audible. You can go ahead.
Thank you, Nanika. So now the session is open for questions. The online participants, they can also ask questions. As we are running out of time, if there is no question, then we can go ahead. And I think Assis Vomik has joined. So I request him to present. Dr. Assis Vomik. I request Assis Vomik to present, please. You are not able yet. We can see you, but your voice is not coming. If you are using headphones, then you have to switch the microphone in your laptop. Your voice is not coming. Okay, we are giving some time to assist for me. Please arrange your system. Now we call Biju Bolpatoki. Biju Bolpatoki, your slides are with us. So you can present now. You need not to show your slides. We are going to running your slides here. I request Dr. Biju Borkotaki to please present. Yeah, you are audible. Yeah, you are. Yes, Dr. Biju. You are audible. Yeah, it's okay, Dr. Biju. You can speak up in few slides. Thank you, Dr. Bhavati. So as we are running out of time, if you have any queries, online participants, if you have any specific queries, if not, then we go ahead with Mitali Purva. The next presenter is Mitali Purva. Yes, visible. Thank you, Mitali, for short and time-bound presentation. Uh, I have only one question because we have time. Uh, like, why have you selected these six species only? Uh, so what is your finding? Should be placed in separate species category or some of them should be merged? Okay, thank you. Thank you. Next participant, Inifa Hassan. Yeah, Candy Girls College. Yeah. Assis Bomi, are you ready to present? Okay, we request Dr. Assis Bomi to wait for some time. Now, I request Inifa Hassan to present, please. Nodi, Nodi, no. Yes, yes, it is visible now. Okay, right. Thank you very much, Dr. Patsa. And now the next speaker, I would like to check Dr. Ashish for me. If you are ready, then you can present now. Dr. Ashish for me. So it seems Ashish for me left the meeting. Yes. The next presenter is Sima Haklari, PH College, Howley. I request Sima Haklari. Sima Haklari, are you present? Yeah, please go ahead with your presentation. Yes, visible. Ma'am, you can skip the ma'am, you can skip the introduction part. Go to the objective directly. Thank you, Dr. Sima. If the online participants have any queries or questions, because we are running out of time. So the next speaker is Tapasri Nath. Tapasri Nath, and you may present. Yes, yes. tapestry to directly come to the conclusion. So as the slides. This so as the slide. Yeah, yeah. Directly come to the conclusion. Yeah, significant factor. Tell us about your significant factors. Significant factors. Thank you. Quantity data. Quantity data. We request to quantity data to present our findings. Yes, that was true. Tell me. Yes, thank you. We have seen your presentation. It's a nice presentation. So as we are running out of time, so many participants are waiting. So thank you. Upaki Deka, are you there? Assis Bhomik sir, are you ready? Now we can present. We can share your presentation from here. You can just present your findings. The slides should, yeah, now slides are visible. I think slides are shared from here. Okay, now slides are visible. So you can present. Dr. Ashish, your voice is not coming. You are not audible. So due to realistic clips, Dr. Ashish could not able to present. So this is the end of this session. And we have 1, 2, 3, 4, 5, 6 presentations in this session, and one is from Ashish for me. So total 7 presenters are present, and 6 have presented their findings. So thank you all the presenters, all the participants, and it's very nice to hear from all of you. Thank you very much. As we are running late, like now 2 o'clock, I think it should be ended around 1:30. So thank you very much. I'm going to give one brief summary to the organizers about the presentations that are on. Thank you.
We would like to thank all the presenters of this session for your wonderful presentation. Now, since we are at the end of this technical session, I would like to request Dr. Neelam Gupta, the convener of the seminar, to kindly give the appreciation certificate to respected chairperson, sir, Dr. Kuldeep Sharma, sir. Excellent.
এই সম্পর্কে